All right. Hello, everybody. My name is Cassandra. I'm a postdoc in Donna Pear's lab at Sloan Kettering. And now we're going to pivot a bit from the spatial stuff and start talking about the chromatin world, and in particular, ways that we've been thinking about improving representation of chromatin for downstream tasks. I'm actually going to start this extremely big picture, and I'm going to talk about this problem that we all know and love, which is if we have some epigenetic information, what can we say about the gene expression of cells? So when you see this slide at the beginning of a talk, chances are the rest of the talk is mostly going to be about what goes into this box. And of course, there's plenty of work on this. I'm sure throughout the conference, we've seen a ton of it. I would say, in my view, the general theme has been, can we go larger? Can we make more sophisticated architectures incorporated into these things and use more and better training data now that we have new regimes like multiome available? But that's actually not what I'm going to talk about today. I'm going to talk about a maybe less well-studied aspect of this problem that I actually think is very important in the analogous problem of text-based prediction tasks, which we all know work quite well. And that is specifically the tokenization of the input. So as Donna loves to say to us, garbage in, garbage out with whatever machine learning approaches you're using. And I would argue that, no matter how sophisticated we're able to make these, we need to have representations of the data going in that are actually going to be meaningful. And so with that in mind, today I'm going to talk about some of our initial ideas to improve these representations for chromatin accessibility data, in particular ATAC-seq data. So let's dive right into that. ATAC-seq, as most of us know, is derived from this assay where we have TN5 enzyme cutting in accessible regions of DNA and then producing this single base pair resolution signal along the genome. So this is a one-dimensional signal. We could use this as sort of the raw form of the data, but what people typically do, and we worked this out quite a long time ago, is that we can really just chunk the genome up into these accessible elements, these cis-regulatory elements, which are typically enhancers and promoters, and then use some summarization of the signal in each of these as input to any downstream tasks. Usually what we're going to do is come up with some embedding and then pass this into whatever we need to do next. So this is probably oversimplifying things a bit, just to be completely clear. There's a lot of complexity to this analysis. And I also would be remiss not to mention that there are approaches which are trying to augment additional, more regulation-focused information into this, such as sequence-level information. All of that is to say that the core of most of the methods we're using today for these analyses is really using this, how accessible is each element representation. And what I want to propose to you today is that this is not actually what the assay is measuring, especially at the single cell level. At the single cell level, we have two molecules of each of these chromosomes, and we are just measuring, where the TN5 enzyme can cut. So what we thought is that really the more appropriate way to think about this data is that it's measuring location that cutting is able to happen. It's really location-based, and this is, of course, constrained by all of these proteins which are bound to the DNA, which are both holding the chromatin open, largely in these promoters and enhancers, but also in smaller regions is actually occluding the ability of TN5 to cut in that position, where you get these kinds of dips, which are called transcription factor footprints. All right, so basically the idea that we have stemming from all of these thoughts was we should treat this data like a probability of TN5 cutting along the genome. So essentially, we're now going to treat these data sets like probability distributions. We, of course, can't measure these things well at the individual cell level because we're only observing one sample, so to speak, from this distribution. But if we kind of look at a population average, either grouping metacells, which I'll talk about, which are kind of high resolution clusters, or maybe even higher or lower resolution, larger clusters of cells, we can get an estimate of the population average of this activity. Okay, so how is this going to work in practice? I'm going to walk you through this toy example here. I have these four, I'm going to use the word histograms from here on out. These could be, let's say, different cis-regulatory elements. They could be across different cells, for instance, and I want to compare them. So I think I've convinced you, hopefully by now, that if I do the standard thing, which would be basically just summing these and looking at how much accessibility was there overall, I'm going to get this, Euclidean distance matrix if I'm just comparing this way, which is not going to differentiate the different patterns I'm seeing here. So our idea, as I've mentioned, is to instead treat these as probability distributions. And in doing so, we're going to use the appropriate distance metrics for probability distributions, which would be Wasserstein distance or optimal transport distance. So just the way you can think about this, if you're not as familiar with this idea, is that when we're computing this distance, we're asking how much work would I have to do to move the mass from one of these distributions to the other. And the key thing here is that I actually care about the location along the genome linearly. So this is biologically relevant because you can think of something like, let's say, a nucleosome sliding along the genome. This is one of the key ways that genes are regulated. We can maybe actually capture this by using a distance metric that's aware of this sort of information. All right, so with this, us single cell people, when we get a distance matrix, we need to do an embedding, of course. So that was the first thing we did. But then I sort of thought, okay, well, I have this embedding, and this is in some sense where these positions fall. You could think of this like the tokenization. This is almost like the idea of word to vec, but very different information, of course. But now each of my peaks has this position in this latent space, which is hopefully more informative about the biology. All right, so how are we going to do this in practice? We can use any method we choose to come up with this embedding. We decided to go for an autoencoder to try to squeeze as much of the signal as we can out of this. So at first pass, we're just going to take each of these histograms, pass them through the encoder. So each histogram gets a single position in this latent space. And then we decode it, and we actually just start with a standard autoencoder stuff L2 loss. So at this point, this has absolutely no Wasserstein or optimal transport level information. So how am I going to use that? We're going to use a trick from a recent paper in our lab, which essentially what it's saying is for any two pairs of points, let's say I have a histogram A and B, the distance between those in latent space in Euclidean distance should be as close as possible to the Wasserstein distance of the original histograms. So you can think of this almost like an MDS, where I'm constraining the positions in this latent space to be similar to the Wasserstein distances of the histograms. Okay, so this is nice, and I tried this. I tried this first on cis regulatory elements, basically centered on transcription factor motifs, where I know the motif, and I got this amazing embedding, and I was like, wow, I have really done something here. I think I've solved it. So then I went and I got even more excited when I looked at Berry centers, which are these histograms that are sort of the average view histogram. I can see a lot of people like, wow, but I'm about to shatter your whole world. So essentially, I showed this to somebody in my lab, and they were like, Cassandra, what about this whole TN5 enzyme bias thing? And I said, yeah, well, that's for the sequence people to deal with. I don't have to deal with that, right? But I said, okay, let me go check it. And very thankfully, the Bonrochter lab just is working on similar ideas. They collected this really nice data set, which is basically an experimentally measured TN5 bias. So basically what they did was they said, let's just have the enzyme cut up naked DNA, and we can now look at what would it look like if I had no actual regulatory signal here, just what does the enzyme like to cut in terms of sequence. So I went and took that model that they developed there and then looked at it in these same regions. And turns out that the vast majority of what I was capturing when I did this was just bias. So, the talk is not over yet, thankfully. We have a few saving graces here. The first thing is that because we can measure this really well, this is good because I can hopefully remove it, right? So I'm actually really thankful for this data set. I think anybody working on sequence level TN5 stuff, you should check this out if you haven't yet. It's really nice. The second thing is that you might say, okay, well, look at this. When I remove it, what's going to be left? Because it looks quite similar. You can take my word for it now, but hopefully as I go through the talk, you'll see that there is, in fact, signal here. You can kind of see it now if you look at some of these flanking regions. There is some structure there which can't be explained by the sequence bias. So in a nutshell, I will spare you of the gory details, but what we did here was essentially come up with a way to try to decompose this out in the autoencoder. So we get this hopefully mostly bias-free signals that we're actually going to use for our Wasserstein embeddings. All right, so that brings me to my results. I'm going to talk about two quick vignettes, the first being more on these TF binding distributions, and then the second being on, teasing this idea of can we do expression prediction very quickly. All of this is going to be on multi-ohm data, and I'm going to very quickly walk you through exactly how I get these histograms because I've kind of glossed over this a little bit. So we're used to looking at these cell by feature matrices where each position or each feature here is a locus. We might have some metadata attached to these, but in our case, our features are now histograms, right? So they're not individual scalars anymore. What I have to do in order to make each of these positions comparable along this axis here is have some common reference frame. So what I've done is centered them on motifs for this upcoming analysis. So basically everything you're seeing is like, let's say minus 100 plus 100 base pairs centered on some motif. And this is going to be really, really sparse. So what I have to do is do some averaging or some summing. I'm going to pseudo bulk over metacells in this case. And then I'm also going to do something in some cases called collapsing, where essentially I'm going to take things that are all associated with the same motif, even though they're different positions, and sum them up. And what this is basically then giving me is sort of the average binding profile genome-wide for that TF. Okay, so finally into the results, I'm going to show you this where I'm now pseudobulking metacells. Each histogram is going to be one motif, basically genome-wide representation for FOXO1. And this is what that data looks like. So you get these really nice representations where each point here, again, is sort of the global FOXO1 binding profile based on shape across the hematopoietic axis. And now we have these points colored by their annotation. You can see how nice the barycenters are once we remove this TN5 bias. We get these really interesting shapes that are driving this. And just to really drive this home, if we let the TN5 bias in, this wouldn't look so good. So we can do things with this. We can, for example, look at diffusion components. This is giving me sort of a continuous axis through this space. And what you can see going from top to bottom here is each of these rows is one of these distributions, is that we're going from kind of having a big dip in the middle to a big peak. And what this is basically looking like to me is a TF footprint for those of you who have looked at something like this. It does correlate with FOXO1 expression. So we thought this was a nice sanity check to say these histogram shapes are actually capturing something biologically meaningful. We can do this for all types of TFs and we get all different types of structures. It turns out that for the most part, about 70% of these are relatively at some moderate level correlated with expression of the TF. In fact, sometimes even more correlated than those which are from other competing methods. And this holds even at pretty high levels. Okay, so I'm going to go into a quick story on focusing a bit more on CTCF, which is a really nice structured TF, so we can get something out beyond just expression. In the next couple slides, what I'm going to show you is, in this case, pseudo-bulking all cells. So you could think of this like a bulk ATAC-seq experiment. Each point I'm embedding is one separate CTCF binding site now. And this is what we get. We get this really kind of funny looking structure with this really strong first diffusion component that clearly separates out between cells, or rather peaks, which are skewed from right to left along the histograms. This is maybe biologically meaningful, I'm not sure, but the one thing we really wanted to do is see, can we capture CTCF binding differences? And we were happy to see that the first diffusion component correlates really nicely with some ground truth ChIP-seq data for CTCF binding. So basically what I've done here is take CTCF ChIP-seq data and look at kind of what's the probability that each of these regions had CTCF binding based on that data. And it does indeed correlate really well with some of the unbiased analysis that we're getting here. We could also look at all different types of ChIP-seq data, and we then incorporated histone marks for active and repressive marks. And then we looked to see that these are actually kind of also correlated with different axes. So what we sort of notice is that there's this kind of like three-dimensional space here. We have really highly clearly bound CTCF sites. We have sites which are having strong active marks, and then we have this corner down here where we have an abundance of repressive marks. So we thought we can kind of use our embedding space to sort of place each position on this three-dimensional axis and then, do a bunch of interesting things. Like we could look at combinatorics, for instance, and say, okay, if I look at sites based on ChIP, which are just bound by CTCF, where do they fall? Where does its cofactor fall? Of course, it's not going to be in CTCF binding sites. But if we look at places where we have co-binding of these two factors, we see potentially shifts in this space. So this is all the kind of things we think we can use and then hopefully now embed more single cell level information onto this to get some information. I will very, very quickly go through this last part. We have this idea of predicting gene expression that I set out at the beginning. So in this case, we're now looking at histograms centered right on promoters on transcription start sites. our embedding looks like this. And then when we overlaid strand, we realized that, okay, we're getting basically which strand the gene is on because the promoter is either upstream or downstream. So we came up with a way to get rid of that using a Gromov-Wasserstein variation that is kind of invariant to the orientation of the histograms. And these embeddings are now much more useful with respect to gene expression information. So this is my last slide. The idea here is that, we have this many models that are really good at doing gene expression prediction for one gene. This is what most people in the field are focusing on. And there's reasons for this because there's so much contextual information that is really hard, I think, to capture. So even very naive methods do quite well at this. Our ultimate goal, what I really hope to be able to accomplish in this project and future projects, is can we come up with a more universal model that really says, like, this is what all of the features need to look like for you to have this level of gene expression. And we're hopeful that we might be able to do something like that with this, because when we look at our latent space, we see that we have really good mixing across genes. And we were able to do a very simple prediction that I think shows that there's some promise here. So just to conclude, I do believe that this, idea we have here is giving us some more deep information than what we could get before from standard ATAC-seq analyses. I just wanted to throw this in there. I threw this in obviously since the conference started, but I liked in John Jumper's talk as he went through this AlphaFold timeline that he mentioned this distogram barrier, and I felt like I could relate to this because I think we have these nice Wasserstein matrices now, but I noticed that my latent spaces are very, very constrained to fit to these. So I do think down the line incorporating all of these other sorts of information will be very helpful. And I'd like to thank everybody in my lab, especially Danielle, who worked really hard on this, and thank you all for your attention.
Thank you so much. Fantastic talk. We have time for a couple of questions.
A very nice talk.
Thank you.
I have a question. You showed you do right now in a supervised way, so you have to use the motif or the TSS to center. If you will do this in an unbiased way, are you exploring this idea?
Yeah. I think that this is like our biggest issue with the method right now is that we don't want to rely on TF motifs. The main challenge is how do we have some kind of common coordinate system? And we're thinking about trying to implement some kind of shift invariant version of Wasserstein distance. So it's sort of we'll automatically do some kind of best alignment. So yeah, that would be the next direction, I think.
Yeah, one last short question.
Thank you. If there is heavy experimental bias, and for instance, if you have a lot of background signal, I'm wondering if the OT will begin to transport the mass to the background signal, and if that affects the results.
Yeah, I haven't systematically evaluated this, but I think the background signal is most likely going to be very TN5 bias driven, which is kind of a good thing in our case because we are regressing that out. So I think if everything's working well, this should hopefully remove the effects of background in that sense. But of course, it's not perfect. So I'm sure that there's some leakage of that. Thank you.
Thank you very much again.